Hi, good afternoon, everyone. Yeah, so we'll get started with the third lecture today. So, hi, I'm Arpit. I'm your TA for this course, I think biology, conducted over in NPTEL. So today we are at live session number 3. So here are some important links as always. So the first one is the session for the live uh, link for the live session, which is this one. Which is ongoing. So yeah, you can get that from here. The second thing is the YouTube link. So the channel where all the live session recordings are being uploaded. And the third one is the Google Drive, which basically has all the PDF forms of all the PPTs that are used in the live sessions. So in week 3, we'll cover all the lectures that were covered. Just a second. Okay. Yeah. Yeah. So we'll today get started with get started with lecture 12 and 13, which were on biomolecules. So um, first I discussed about so first we took a look at the periodic table, and here all the elements are basically colored based on if they are essential to human beings or not. So purple indicates that the element is essential for human beings. Green indicates that it is suggested to be essential for human beings, and gray is for non-essential elements for humans. Here we see that so some of the most essential elements for humans are this hydrogen, carbon, nitrogen, oxygen, and phosphorus. So these kind of, as we look forward in the lecture, these basically help form the backbone of all the structure and all the molecules, biomolecules that are used in the human cells, right? The organic backbone. And if we look at this left-hand side corner, we see that we have metals such as sodium, magnesium, calcium and potassium, which are basically present in their ionic forms, and they basically help. Basically, they act as cofactors, or they bind to different biomolecules and make them stable, or catalyze certain reactions, and so on. And we also have elements such as iron, cobalt, manganese, and all, which are essential. So iron is one element which is present in hemoglobin, the biomolecule which basically helps us carry oxygen through our blood. Mm. So yeah, so these are the essential elements for life, for human life. So two most so of all the elements and compounds that are present, water is probably the most common constituent of any form of life that we have on Earth. So mm, basically the polarity of the water molecule. So since oxygen is more negative than hydrogen and much more bigger, what it does is it basically pulls a lot of the electron cloud that is present in the molecule towards itself, where delta negative charge is generated on the oxygen molecule, oxygen uh, atom as we see here, and on the hydrogen atom, we have a slight amount of positive charge. And due to the presence of this polarity, this positive, slight positive and negative charge, water can form what are called hydrogen bonds. So these are weak associations between water molecules inside water, and basically that gives some sort of a loose structure or some sort of viscosity and fluidity are things that are basically influenced by these. Then we have carbon, which is mostly present in the body in the form of hydrocarbons. So they may exist as linear carbon chains, carbon rings, or combinations of both. And we have basically individual carbon to carbon bonds that are present in these molecules, which may be a single bond, a double bond, or a triple covalent bond. So, for example, here we see that ethane has a single bond between two carbon molecules, carbon atoms, whereas ethene has a double bond. Right. And again, we have another molecule C2H2, Chilean, I guess, which basically has a triple covalent bond between two carbon molecules. Right. So the covalent bond also affects the structure of the molecule. So for example, you see if carbon has a single bond, it forms a tetrahedral structure. But if carbon has a double bond, it tends to form a planar structure. So in biology, there are basically two very common reactions that are found in almost all places. So the first thing is a synthesis reaction or a dehydration reaction. So it's called a synthesis reaction because a polymer chain is synthesized or molecules are combining to form new molecules. And it's also called a dehydration reaction because one molecule of water is being thrown off. So basically, let's say you have these two glucose molecules which are attaching, and this have water. So this OH and H group basically dissociate to form water, and there's a bond that is forming between these two molecules. Similarly, hydrolysis is the opposite of a synthesis reaction. Hydrolysis is basically a breakdown reaction. So what is happening is addition of water is leading to lysis of the bonds. So lysis is breakage. So you add water and basically this dimer breaks down into its individual monomers. So again, the H goes in to one of the oxygen. So the X binds here to one of the carbon and the H binds here in one of the carbons and the OH binds to the other. Right. So uh, these are the four building blocks of the cell that are present in each and every cells. So these are basically sugars, fatty acids, amino acids, and nucleotides. And these basically polymerize. So individual units of sugars combine by these different bonds formations via these different reactions to basically give rise to polysaccharides, which are basically polymers of sugars, different kinds of sugars. Then we have fatty acids, which polymerize to give us fats, lipids. So these things are mostly used in cell membranes and nuclear membranes and so on. Next, we have amino acids, which polymerize to give us proteins. And we have nucleotides, which basically polymerize to give us nucleic acids, such as DNA and RNA. Here we see an example of what proportion of a cell is what component. So you see that most of the cell is composed of protein, then we have RNA, then we have DNA, then we have lipids, glycogen, which is the polysaccharide, and other things such as peptidoglycans, polyamines, and so on. But the thing to note here is that proteins are the most abundant thing in a human cell, the macromolecule in a human cell. And then the other trend we see here is that RNA is much more abundant than DNA in a cell. Right. So here is again a tabular form of the whole thing. So here we are comparing basically the prokaryotic cell and the eukaryotic cell. So E. coli versus a mammalian cell. What is the percentage of different components? So we see that again, like both the cells are almost made up of 70% of water. Then we have 1% of inorganic ions such as sodium, potassium, magnesium, calcium, and so on. And then we have some small metabolites, so small metabolites and other products that are produced during the process of metabolism or energy generation. Then we have proteins, which make up a large chunk of the cell, followed by RNA and DNA. And lipids and all, phospholipids, polysaccharides, they fall somewhere in between. Right. So in biomolecules, we talk about different biomolecules. So the first one we talk about is DNA. So a DNA molecule is basically made up of three things. So the first thing is the sugar here. So this is called a deoxyribose sugar. Uh, then we have this thing called a nitrogen base. In this case, it's a thymine, then a adenine, a guanine, and a cytosine. So these are different nitrogenous bases that are attached to one carbon of the sugar base. So we have a sugar, we have nitrogen base, and the third component is this thing called a phosphodiester. So basically, we have a phosphate group in each of these sugar molecules, which gets attached to the, so the carbons here are numbered. So the numbering starts from here. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So these are the five uh, carbons. So the sugar is deoxyribose for DNA and ribose for RNA. So deoxyribose is the five carbon sugar. So it has five carbon atoms, and these are numbered as shown here. So what is happening is in the 5' carbon, so in the 5' carbon, we have a phosphate group, which basically attaches to the uh, oxygen atom, the OH group, that is the O end of the next sugar. So basically the 5' end attaches to the 3' end of another sugar molecule. And that's how this phosphodiester bond is formed. And that's how this basically structure gets elongated to form the TN. And here what we have is basically, so this is the phosphodiester bond and the basic structure of how its single DNA strand is formed. And here we show that nucleotides also serve as basically, so they serve as information storage units because they are present in the DNA and they are storing information, and they are also serving as energy carriers. So here we see that we have these ATP molecule, which is basically adenine attached to a ribose sugar with three phosphates. And this is basically the energy carrier that we have in a cell, breakdown of ATP into ADP. So basically leads to release of energy because of breakage of this phosphate bond. And that basically powers the cell. And again, like if you have these two components, IADP and inorganic phosphate, or phosphate group, you can basically provide energy to form a ATP molecule. So basically during the process of photosynthesis or during the process of, let's say, mitochondria like respiration, uh, ATP is produced, which basically stores the energy that is generated in a chemical form. And then later, when that energy is needed, the ATP molecule hydrolyzes to basically give out the energy. So here we see that, so as we said, so the DNA strand that we have here runs from basically, so at one end it will have a 5' end where the 5' carbon is basically free, and on the other end it will have a 3' end where the 3' prime carbon is basically free to form a bond, and therefore the strand of a DNA can have a sequence between this 5' and a 3' end. So what happens in DNA is that we have two strands that are running anti-parallel to each other. So one is running from 5' to 3'. So that this one, which goes from 5' to 3', and the other strand which is bound to it runs from 3' to 5'. So complementary, anti-parallel to each other. Right? They're moving in, they're running in opposite directions. And then we have these different nitrogen bases. So what happens is we have two strands, each of which have a sugar phosphate backbone. So it's made up of the sugar bound by phosphodiester phosphate bond. And then what we have here are the bases. So when the bases, basically when they're in close proximity in the DNA, they basically form hydrogen bonds within each other. So adenine and thymine form 2 hydrogen bonds, whereas cytosine and guanine form 3 hydrogen bonds. And that's how the DNA molecule is bound into a, basically we have 2 strands of DNA with basically different bases, which are then bound to each other using hydrogen bonds. And then they wrap around into this helical structure. Uh, so this helical structure has different properties. So the width of it is around 2 nanometers. Then we have a major groove and we have a minor groove. Here again, we can see that this is the major groove and this is the minor groove, minor, major, right? And we see that the strands are running in antiparallel directions, and we have hydrogen bonding within each base pair. And if you see, look at it closely, so the nitrogen bases are basically these circular structures which have a delocalized electron pair. So kind of similar to what we have in benzene, right? So this delocalized electron cloud, basically what happens is consecutive uh, nitrogen base pairs basically stack on top of each other, and that pi-pi stacking that we call. So the pi-pi stacking is basically the delocalized electron cloud that is present on top of this. So that stacking basically yields structural stability to the DNA, right? Along with the hydrogen bonding and the sugar phosphate backbone and the coiling. Right. Next, we talk about RNA. So RNA is generally a single-stranded molecule unlike DNA. And so there are certain differences between DNA and RNA. As I said before, RNA contains ribose sugar, whereas DNA contains deoxyribose sugar. So in DNA, if you notice in the 2' carbon here, we have just a hydrogen, whereas in RNA, we have a OH group here. So because that oxygen is missing, it is called a deoxyribose sugar, whereas in RNA, it's called a ribose sugar. So RNA also forms phosphodiester bonds to link to each other. And then again in the 1' carbon binds to a nitrogen base. And in RNA, we have uracil, which is another nitrogenous base present in place of thymine. So they're very similar in structure, except for this methyl group here. Right. And the rest of them are same, adenine, cytosine, and guanine. So what RNA can do, since it's a single strand, what can happen is it can fold on itself and basically bind to certain places which are complementary to it. So as I mentioned before that uh, C basically binds to G using a triple hydrogen bond. A generally binds to basically T or U using a double hydrogen bond. So basically, if you have these base pairs opposite to each other, they can still form the hydrogen bonds. And that's how you get these hairpin-like structures in an RNA, or you get a 3D structure in RNA as shown here. So here you see a 3D structure of a RNA strand. Note how it's like uh, coiling and basically folding on itself and basically forming hydrogen bonds and hairpin loops. Right. Next, the next important macromolecule is basically protein. So we talk about protein. So proteins are made up of smaller structures called amino acids. So we have 20 amino acids. So they have a general structure like this. So basically you have a carbon atom, which is bound to a hydrogen atom, a carboxyl group, COOH, and a amine, NH2. And then we have this R subgroup, which can basically vary. So based on what is present here, we have different kinds of amino acids. If R is equal to H or a single hydrogen atom, we have something called glycine. And if R is basically a methyl group, we have something called alanine and so on. Right. So here you can see the structure where you see this is the nitrogen atom, this is the carbon atom, The red ones are the oxygen atom and the white ones are the hydrogen atom. So this is basically glycine, where the we have a NH2 group here, we have a COH group here, we have a hydrogen here, and we have another hydrogen. So R is equal to H here. And here we have alanine, where we have a carboxyl group, we have a NH2 group, we have a hydrogen, and we have a CH3. So R here is equal to CH3. Right. So we have these individual amino acids. So based on the side group that they have, it can be non-polar. For example, glycine and alanine are non-polar because CH3 and H are both non-polar. But there can be other amino acids which have positive side chains or negative side chains, which basically make it polar and positive or negatively charged. Right. Next, what we see here, again, using a dehydration reaction or a synthesis reaction, what is happening is one water molecule is taken out, and we have the formation of a peptide bond between these two amino acids. So what we'll do here is use it so that it's easy to read. Okay, so yeah, basically what is happening here is this part, which is basically one oxygen molecule and two hydrogen molecules. Basically, they separate and form water, and they go out. And this part joins to this part. What we have is called a peptide bond. So the carboxyl group here attaches to the amine group here. And again, this carboxyl group can attach to another amine group. This amine group can attach to another carboxyl group. And that's how proteins are formed from amino acids by the formation of peptide bonds. So basically, two proteins combine and a water molecule goes out. And two amino acids combine and a water molecule goes out. And we have a basically dipeptide, tripeptide, and so on, polypeptide. Polypeptide chains are called proteins. Now, this is the first order of thing. So a protein will have a primary structure, which is basically the sequence of amino acids that are present in the protein. So for example, we can have a protein primary structure such as A, G, K, L, P. So these are the 5 amino acids that come together to form this protein. So as I told, there are 20 amino acids. They are denoted by these 3-lettered abbreviations or these 1-lettered abbreviations. So aspartic acid becomes ASP, glutamic acid becomes GLU, glycine becomes GLY, and so on. And we have a single digit code also. So this is how we can write a primary structure of a small protein, let's say. Let's say there are just 5 nucleotides. There are 5 amino acids. Then we have this protein, and this is the primary structure of that protein. Now, what happens is we have the formation of something called a secondary structure. So what happens here is as the protein or the polypeptide chain wraps around like this sometimes, what happens, there are hydrogen bonds forming between these nearby carboxyl and amine groups. And basically that leads to a helical structure in some cases. So we have what are called alpha helices in proteins. Similarly, if the protein polypeptide backbone does not fold like that, but it comes into contact with, let's say, another polypeptide backbone and forms hydrogen bond like this between them, we'll have something called a sheet structure or a beta sheet. So this is the secondary structure. So this is the primary structure of a protein, and this is a secondary structure of a protein. Now, once we have a secondary structure, what can happen is we can have bonds in between different parts of the polypeptide chain. So sometimes the polypeptide chain will be much larger, and there can be bonds between different parts of the polypeptide chain that lead to a tertiary structure. So for example, here we have an alpha helix, which is our secondary structure. We have a beta sheet. And basically, we have some hydrophobic interaction here, which is binding this alpha helix to this part of the protein. Right. Then we have some hydrophilic interaction, which is basically binding this part of the beta sheet to, let's say, water molecules that are present in the cell cytoplasm and so on. And we can have salt bridges where positive and negative ions basically come together to form a salt bridge. We can have random coils in the protein. We can have covalent bonds, such as these sulfur disulfide bonds. Right. We have hydrogen bonding and we have metal ion coordination. So these metal ions will sometimes come in between proteins. So they basically draw together different negatively charged parts of the protein, and they basically help it form a tertiary structure. So that's why sometimes these metal ions are very essential for us. Uh, Things such as iron, magnesium, calcium, and potassium, and so on. Right. And we can have hydrophilic interactions, hydrophobic interactions, and so on. So this is basically a tertiary structure of a protein. So we have a primary structure, which is basically the sequence of amino acids. We have a secondary structure, which is basically alpha helices or beta sheets, which are formed by weak hydrogen bonding. Mm. Then we have tertiary structure, which is more rigid and formed by other kind of interactions also. Then we have something called a quaternary structure. So a protein is generally made up of different domains. So basically, we have these different domains that are present in a given protein. For example, this is the SH3 domain, this is the SH2 domain. These basically contain some beta sheets. Then we have a small kinase domain here, which has a lot of beta sheets. And we have a large kinase domain, which has a lot of alpha helices. Right. So this whole thing is one strand of protein. It's just this part has a different tertiary structure. This part has a different tertiary structure. This part has a different structure. And this part has a different tertiary structure. And these all are based on the virtue of that tertiary structure are coming together to basically form a protein molecule like this. So this is now called the quaternary structure. So sometimes some proteins are basically made up of many domains that are similar to each other. For example, this four fibronectin type 3 domains basically mix to form a longer chain, a longer fibronectin fiber. Right. So what is the importance of this? What is the use of this? And basically, why should we know about this? So it's very difficult for us to basically predict the uh, functioning of a protein based only on the primary structure or the sequence of the protein. So, for example, if we have a DNA sequence, we can exactly say that how a corresponding DNA strand or how a corresponding RNA that is formed of the DNA would look like. But and basically, so we can say, okay, this DNA makes this RNA, so that's why this has this function, and so on and so on. But in the case of a protein, we cannot just like look at the sequence and basically say what function it's performing and so on. But we need to look at is also the structure of the protein. So here is an example showing chymotrypsin and elastase, which are basically uh, proteases that are created in our pancreas, and they work at an optimal pH of 8.0. So here we see that even though both have different primary structures, both have different strands, different amino acid sequences, they are in a way forming similar structures because they are performing a similar function. So the tertiary structure of both of these are very similar. Right. Only 40% of their primary structure is similar, but their tertiary structure is very similar, including this site where basically the processing of things would be happening. Right, the active site. We also see this thing in evolution. So where we see that there are certain proteins that are very essential for functioning and so on. So for example, uh, these proteins, so the yeast alpha 2 protein and the Drosophila engrel protein, which are basically DNA binding um, domains, which are called homeodomains. So these are separated by more than 1 billion years of evolution in these two organisms, Drosophila and yeast. Drosophila, which is a fruit fly, and yeast, which is basically a microbe. So what we see here is an amazing similarity between both of them. Right. So even in the sequence, we have like similar kind, positively and negatively charged, similar kind of amino acids that are present, and a similar structure is something that we can observe here. So proteins that are highly conserved, proteins that are highly essential for certain kinds of very important tasks generally tend to have similar kind of structure that does not change over evolutionary time that easily. Next, going above the tertiary level, we have what is called the quaternary structure of a protein. So as we talked about here before, here we can see that the neuraminidase basically is a enzyme that basically is a tetramer of these four polypeptide chains. So the same basically uh, monomer, the same peptide chain basically binds 4 times to form a molecule like this. And we see that there are like similar red, yellow, blue, and green beta sheets present. It's just rotated in a way so that they can align and bind to each other. Here we see a comparison of the different kinds of protein molecules that we can have in a cell, starting from very small protein molecules or domains such as the SH2 domain or the lysozyme, to very large proteins such as aspartate and basically deoxyribonuclease and alcohol dehydrogenase and very long peptide proteins, long protein structures such as collagen and so on. Right. So here is again another example showing what are the different classes of proteins that are present in the human cell. So around 23-24% of these proteins that are found are not classified, but the rest of them are classified into some kind of class based on the function that they're providing. For example, proteins can be storage proteins, can be providing structure as structural proteins. They can be surfactants or basically cytoskeletal proteins, which provide uh, structure to the cytoskeleton of the cell and so on. Right. So that was all that was taught in the first lecture. Now we move on to the second lecture, again on biomolecules. Does anyone have any questions or doubts till now? Please feel free to unmute yourselves or ask questions in the chat box. Okay. So in the next lecture, what we looked at was basically polysaccharides. So polysaccharides are basically polymers of sugars. So the simplest sugar that we can think of here is glucose. So glucose has these different forms. So it has a basically straight line form, and it has two ring forms. So this is alpha glucose and this is beta glucose. So this alpha and beta is based on the presence of the hydroxyl group on the 1' carbon. So if it is present opposite to the CHOH group, it's known as alpha glucose. If it is present on the same plane, it's known as beta glucose. So alpha glucose can form both 1,4 glycosidic and 1,6 glycosidic bonds. So what basically happens is in 1,4 glycosidic bond, this carbon 1 combines with carbon 4 in another uh, glucose molecule. So these two link together and a water molecule comes out to basically form a bond here. And these links are basically called 1,4 glycosidic bonds or 1,4 glycosidic links. Then we have 1,6 glycosidic link, which is basically uh, 1' carbon binding to 6' carbon. Right. And starch is basically made up of 1,4 linkage of alpha glucose monomers, and cellulose is made up of 1,4 linkage of beta glucose monomers. So here you can see that in starch, all the OH groups are on the same side, whereas in cellulose, they keep alternating. So one interesting question to think now is, why can we digest starch but not cellulose? Because both of them are made of the same kind of bonds. Is that just that one is made up of alpha glucose, one is made up of beta glucose monomers, right? So we think about the question. We'll now go and look at the more about the structure of these molecules. So starch basically can be amylose or amylopectin. Amylose basically has only 1,4 glycosidic bonds, so it forms a straight chain, and this 1,6 glycosidic bond basically leads to a branching that's happening. Because once you form a bond here, it starts from here. The next chain starts from here. It's not linear anymore. Right. So starch can have 1,4 or 1,6 glycosidic bonds to form amylose or amylopectin. Glycogen has a lot of these branching. So starch is generally present in plant food and so on. Glycogen is generally present in, for example, human cells and so on. These are used for temporary storage of glucose molecules. So why they generally tend to have more 1,6 glycosidic bonds than 1,4 glycosidic bonds, and this is the case because a lot a lot of 1,6 glycosidic bonds leads to more branching. More branching leads to more surface area, so that when glucose is required by the body, these glycogen molecules can be broken down efficiently because they have larger surface area where more number of enzymes can act to break it down. Right. And yeah, so that's it. And cellulose, what happens is they have 1,4 glycosidic bonds, and then these alternate OH bonds basically bind to uh, different strands to form a more rigid structure. So that's why it's easier to digest starch than it is to digest cellulose, because cellulose has a more rigid structure because of the beta glucose molecules that are present. They are basically forming these interlocking patterns, whereas the starch protein, whereas starch, where only alpha glucose molecules are present, don't form this rigid structure. Okay. Next, we have glycoaminos glycosaminoglycans, which are also called as mucopolysaccharides. So these include generally keratin, sulfate, heparin, hyaluronic acid, and chondroitin sulfate. So these basically contain a repeating unit of a disaccharide shown here, which has at least one negatively charged residue. So what happens is that we have a negatively charged residue in these disaccharides, which basically results in a structured polymer, which basically has a lot of anions, and therefore it has more interactions with water. It is more hydrophilic. So the negatively charged part generally basically interacts more with water to give it a slimy feel. So these molecules are generally found in places such as mucus, snot from your nose, and so on, and in synovial fluid, which is present in joints to basically lubricate the joints. So some of these things, such as chondroitin sulfate and so on, I think are given as um, medicine for people suffering from arthritis or other kinds of joint diseases. Right. Next biomolecule we have is lipid. So lipids basically mm, are these molecules. I think there's some issue with this slide, so the end carboxyl group is not visible. I'll update it before posting. But basically, what happens is again, there's lipids from things such as triglycerides. So triglycerides have three lipid molecules stuck together. Then we have these so these are long chain fatty acids and lipids. Then we have these phospholipids, which have also a phosphodiester bond or a phosphate group. So basically, these phospholipids are also present. So these glycerides and phospholipids are generally present in cell membranes, nuclear membranes, and so on. Then we have this molecule called sterol, which is basically something like this. It's basically a ring-like structure, ring-like lipid. The most famous example is cholesterol, which serves as a starting point for many hormones in humans. Right. Here are some other molecules and a comparative analysis of their size. So here we have like glycerophospholipid, we have glycerolipid, so glycerine and lipid combined together, then glyceryl lipid combined together with a phosphate molecule, then sphingolipid, fatty acid, sacrolipid. Sacrolipid basically has a sugar molecule in that, and so on. So these different kinds of polymers can be formed that carry out different kinds of functions in the cell. Here we have shown basically how the cell membrane is formed out of lipids. So lipids have this basically structure where basically there's a charged end and there's a uh, non-charged, non-polar end, hydrocarbon end. So this hydrocarbon end is generally hydrophobic, whereas the charged end is generally hydrophilic. So what happens is basically we have this bilayer sheet where the hydrophobic ends are together and the hydrophilic ends are on the opposite side. And basically they are in contact with the water, the cell cytoplasm, extracellular matrix, and so on. Right. And so these phospholipids can basically form things called as micelles, which are basically a single layer of these, or we can form things such as liposomes, vesicles, and so on. So which are basically different, what do you say, vesicles or different compartments or different uh, organelles that are present in the cell. Right. And again, this cell membrane has a lot of proteins and a lot of other lipid membranes that are attached to it, such as this glycolipid, this glycoprotein. So this protein that is attached to a carbohydrate, glycolipid, which is a lipid attached to a carbohydrate. So then we have these other uh, lipid molecules such as cholesterol and all, which are also present in the membrane, which also influence the fluidity of the membrane. Then we have these channel proteins, which basically form channels between the membranes, and integral membrane proteins, which are integrated into the membrane and are present there, and so on. Here is again another view of the cell membrane, where we see that this is the external side, this is the internal side, and these are basically GPA anchored proteins that can flow inside here. Then we have cholesterol, which determines the fluidity of the membrane, as I said before. Then we have saturated and unsaturated lipids, which also again determine the fluidity. It's the influence of fluidity. And then here in the intercellular side, we have these actin fibers, which are present in the cell membrane. So this whole complex is basically how the cell membrane is formed. Now, this is again a 3D representation of the same thing. Here we see a molecular model of a synaptic vesicle. So a vesicle basically, um, yeah, is again like a membrane, is a cell organelle which has a membrane, right? Which is used for generally transporting stuff or carrying things from one part of the other in the cell. Towards the end, we talked about chemical synthesis of hormones. So we talked that this hormone called cortisol was known to be a good treatment for arthritis. So if you have arthritis in olden times, if they give you cortisone, you're basically you're unable to walk in the beginning or move your hands and joints, and then after having this medicine, you can walk and you can basically have reduced pain in your joints and so on. So the way it was produced before was that bile salt was used as a precursor to produce this uh, hormone called cortisol. So basically what people would do is basically what companies would do is basically they have slaughterhouses from where they take bile salts from uh, animals that are slaughtered and basically use that to create cortisone. But that's a very expensive process because not a lot of cortisone is generated in that way. Not a high yield is generated in that way. And also you need a lot of bile salts. Then this company called Syntex came up with another method of uh, basically chemical synthesis of cortisone using this uh, plant sterol. diosgenin, which is found in the Mexican yam plant, which is shown here. So what they found out is that using this plant sterol, which has a slightly similar structure to cortisone, we can make cortisone. And basically they made cortisone and they sold it. And as we see here, that cortisone is also similar to other hormones that are found in the body, such as progesterone. So eventually people develop ways to create progesterone. And basically these are now sold as birth control pills and so on. It. So that's it about the first two lectures on biomolecules. Does anyone have any questions or doubts? Please feel free to unmute yourself or send a question on the chat box. Okay. In the lecture, we talked about central dogma. So here is basically Francis Crick, who is basically presenting his idea of the central dogma. So here behind you can see some reference of what the central dogma is. We'll talk about it now. So first we'll talk about what was known in 1957 when we proposed this idea. What was known was that DNA is the molecule of heredity as established by experiments carried out by Avery and Hershey Chase. Then DNA structure was explained by Watson and Crick. Then Stahl gave a method for self-replication of the DNA molecule. Then people were uh, basically aware of the presence of microsomes, but they didn't know that they are ribosomes. Like now they are basically called ribosomes, but people didn't know what function they do perform. mRNA was not known. mRNA was came to be known in 1961. tRNA was also not known, but Crick basically gave this adapter hypothesis, which is basically the uh, part of the central dogma, which basically says that there's a molecule which should be able to bind to a RNA and generate a protein. So there needs to be an adapter molecule which can basically translate. Uh, so basically, this is what the central dogma is. This is what Watson proposed is that DNA self-replicates, as we know, right? Then DNA produces RNA. So DNA produces RNA through a process called transcription. And RNA produces protein through a process called translation. Right. So basically, this translation needs a molecule which can attach to both RNA and protein and amino acids. So basically, this is the adapter molecule that Crick proposed, which is now known as the tRNA. So basically, this is what he proposed. He said that DNA self-replicates and DNA can form RNA, and RNA can also self-replicate, and RNA can form protein. DNA can also may form protein, but he said that never protein can form protein, or protein can form RNA, or protein can form DNA. Right. So this bottom diagram is never true. This top diagram may or may not happen. Right. It may or may not happen. It happens. So that is the central dogma. So what we know now is basically from what he said, DNA to DNA is allowed. It is called self-replication. DNA to RNA is called transcription. RNA to protein is called translation. RNA to DNA is done in certain retroviruses by the use of enzymes such as reverse transcriptase. Then we have RNA to RNA, which is RNA replication that is shown to be there in coronavirus and tobacco mosaic virus. DNA to protein, there's no evidence, but Watson said that, Crick said that that is possible, but there's no evidence of it as of now. Protein to DNA and RNA is not allowed and not observed, and protein to protein is also not allowed and not observed in nature. So this is basically the central dogma that we talk about. DNA forms RNA, forms protein, DNA can self-regulate, RNA can self-regulate, and DNA. So this is what um, Crick proposed, right? So the question then arises, if DNA can perform like make protein, why not do that directly? Why do we need a RNA in between? Right. At least for eukaryotic cells where the nuclear component, the basically genetic component is made up of DNA. Right. So here is a straightforward benefit of why having a molecule like RNA can help. So for example, you have a copy of gene A and you have a copy of gene B. So the amount of protein that they produce can vary differently, can vary a lot based on the amount of RNAs that are produced from this DNA. So since you have just one copy of gene A and one copy of gene B, producing protein directly from them will give you equal amounts of protein, right, for uh, each time you go through it, right? Whereas if you create multiple mRNAs or messenger RNAs, you can basically translate that into a lot of proteins. Whereas if you have just a single RNA, you can translate that into less protein. So you can have a level of control here. Right. The other thing is your DNA does not get damaged because translation is a very regular process because our body needs a lot of protein, a lot of macromolecules to function. So basically, opening up the DNA every now and then to create proteins is also very difficult. That's why it provides an additional layer of security and stability to the DNA, right? So that it's not destroyed easily. Right. So this is this figure here shows the transcription of two genes as observed under the electron microscope. We can see that basically the transcription goes from here to here because these strands are larger and these strands are smaller. So the transcription is carried out by a enzyme called RNA polymerase. So what this RNA polymerase does is it has a group where basically it can attach to DNA, then it can unwind uh, or open the DNA, and then based on the uh, nitrogen base pairs that are present on the DNA, it synthesizes a RNA transcript of the same, and then basically drops it out and moves on to the next part of the DNA, and so on. Right. And here the important thing to note is that there's a direction of transcription. So basically, the new RNA that is transcribed will always be transcribed in the 5' to 3' direction. Right. So here you see that the 5' end has formed, whereas the 3' end will form eventually when the protein is when the mRNA is fully transcribed. Right. So this is basically the process. So we have something called a sigma factor, which binds to the RNA polymerase, which helps in the binding of the DNA molecule to the RNA polymerase. Then the sigma factor moves out, and basically we have opening of the DNA chain. We have formation of the RNA, then opening of the DNA, and basically formation of the full RNA. Then the RNA goes out. So note here that this RNA will be formed from the 5' end to the 3' end. Right. And the strand that was used to produce this would be used in the 3' to 5' model. So now, if you see here, we have different kinds of genes. We see that since DNA has two strands, both the strands can be used to transcribe RNAs. So for example, you have gene A on this strand. So the direction of transcription will be this way. So going from the 3' end to 5' end on gene A to produce a 5' to 3' RNA. Then again for gene D, gene E, it will again be in the same direction. But on the other strand, which runs anti-parallel, we see that this will be 3', this will be 5'. So the direction of basically the gene uh, transcription here will be opposite on the other strand. So prokaryotic cells have one RNA polymerase. Eukaryotic cells have three kinds of RNA polymerase. So RNA polymerase 1 is basically used in transcribing the rRNA genes. So rRNA is basically ribosomal RNA that is present in the ribosome. RNA polymerase 2 is basically used for widespread protein coding genes for transcription of them. And then basically it also transcribes some small nuclear RNA or snRNA. Then we have RNA polymerase 3, which basically helps transcribe tRNA genes and some rRNA genes and some snRNA genes. Small nuclear RNA, ribosomal RNA. But so the bacterial RNA polymerase is similar to the RNA polymerase 2 that is found in the eukaryotic cell. Here we see that the eukaryotic polymerase is larger than the bacterial enzyme. Right, it has 12 subunits instead of 5. All the additional regions are shown here in the gray color. The blue spheres inside basically represent, uh, for example, here, basically represent zinc atoms that serve as structural components of these polymerases. Then we have, then we have red spheres somewhere, which are basically where magnesium atoms are present at the active site where polymerization is taking place. And then we have, so the rest of the structure that's not gray is basically similar in both bacterial RNA polymerase and eukaryotic RNA polymerase too. And this is because both of them do the same function. And this protein has been conserved over millions of years over evolutionary time, right? Because it performs a very crucial function in the cell. Now, okay. So the other thing to note is that basically human genes, unlike uh, not human genes, eukaryotic genes, like prokaryotic genes, have something called exons and introns. So exons are actually parts which code for a protein sequence, and introns are parts of the DNA that are transcribed but that do not code for a protein sequence. So what happens is basically, so there's the example of two human genes showing the arrangement of introns and exons. The first human beta globin gene has three exons, whereas the human factor VIII gene, which synthesizes a protein molecule called factor VIII, which helps in blood clotting and all, has around 26 exons, which are spread over the DNA like this. So this differentiation of intron and exon basically gives rise to, so what basically happens is after mRNA is transcribed in a eukaryotic cell, it has to get spliced. So the introns need to be removed before it can basically form a proper mRNA or a messenger RNA, which can basically give rise to a protein. So here what the benefit of this intron exon framework is that by the process of transcription and alternative splicing, what we can do is we can create variations of the same protein by just removing or adding certain exons into the mRNA message. For example, here is a gene that codes for alpha tropomyosin, which is basically a muscle cell, right? Muscle protein. So by alternative splicing and transcription, what can happen is the same gene in eukaryotes can give rise to multiple mRNA fragments or messenger RNA fragments, which basically lead to different kinds of muscle cells, or different kinds of muscle proteins. For example, fibroblast or smooth muscle or striated muscles. Right. So that's one benefit of having an intron exon system. So what are the implications of all this? So the implication is that RNA can basically for RNA can basically act as an enzyme. So these are called RNzymes. Then we have this other claim that the world was initially made up of RNA and DNA came later on. And that's why RNA is the only molecule that, as far as we can see, can translate protein. Right, translate protein. The presence of introns and exons leads to splicing. and generation of many variants. So one gene can give rise to many different variants of the protein. Then the next thing we know is that since eukaryotes have a large amount of introns and uh, less exons and so on, so the percentage of non-coding DNA is a lot in eukaryotes. Only 1.5 to 2% of the whole genome codes for protein, whereas we also know that two-thirds of the genome is transcribed for RNA. Right. So we have these RNA which are produced by the genome, but which are not basically coded into proteins. So what are these different kinds of RNA that we have? So the first thing is mRNA or messenger RNA, which codes for proteins. Next is rRNAs or ribosomal RNA, which form the basic structure of the ribosome and basically catalyzes protein synthesis. We have tRNA or transfer RNA, which basically helps us synthesize proteins by acting as an adapter between mRNA and amino acid. We have snRNA or small nuclear RNAs, which function in a variety of nuclear processes, especially in splicing of pre-mRNA. We have snRNA or small nuclear RNAs, which are used to process and chemically modify rRNAs. And we have other non-coding RNAs, which function in diverse cellular processes, including chromosome inactivation, transport of proteins into the ER and telomere synthesis, and so on. So what happens in the process of translation? We have an RNA strand. Then basically we have some enzyme that's binding to it and producing a protein strand. So how does the RNA thing get? So from DNA to RNA is very simple because DNA is formed of ATGC. RNA has a UGC. So instead of T, you have U. But again, like if you have a strand of DNA, you can basically have a corresponding strand of RNA that you can directly transcript. So A here will become U, T here will become G, G will become C, C will become G, and so on. So this will be a RNA that is produced from the DNA strand. Right. But how do we translate this ATGC into proteins? So this is called the genetic code. So we have basically three base pairs that code for a given protein. So we have this three base pairs. So the first letter is here. So the first letter of these are U, the second letter is U, and the third letter is U here. So this becomes UUU, this becomes UUC, UUA, CCC, GAG, and so on. And these are basically the three-letter codes of different amino acids that are produced by using these codons. So for example, these two codons produce histone, these produce leucine, these produce serine, these produce tyrosine, and so on. We have something called AUG, which produces methionine, but is also the start codon. So basically, if translation machinery encounters AUG, that means this is where you should start reading. So AUG and whatever follows is basically the genetic code, and basically you can start translating proteins from there. Then we have these three codons, UUA, UAG, and UGA, which basically are the stop codons. So these tell the translation machinery that stop now, the protein is created. Right. So there are various characteristics of this genetic code. So the first thing is it's universal. So all organisms on the earth use the same code. Right. Next thing is it's commaless. So once you start with AUG, till you encounter a stop codon, you cannot stop anywhere. So whatever is there will be transcribed and translated into a protein or a peptide chain. Right. The third thing is non-overlapping. So if you have something like this, So this will be one codon, this will be the second, this will be the third. You can't have this, this, this. So this is not possible. This is correct. It's not overlapping. It's non-ambiguous. So basically, each of them will lead to a single protein, a single amino acid that is to be attached. It is degenerate or redundant. So many of the codons code for the same amino acid. So that's why it's degenerate or redundant. And there's directionality. So as we talked about it before, there's a directionality in which proteins are produced. Similar to there's a directionality in which RNA is produced and so on. So now we come to the interesting molecule called tRNA. So this is basically a tRNA molecule. This is a schematic structure, and this is basically a another schematic structure showing different parts. So we have the T arm, the D arm, and we have basically an anticodon arm, which contains the anticodon. So anticodon is basically what binds to the DNA. So binds to the mRNA. So mRNA has UCC, let's say. This will bind to AGG. Right. And in tRNA, what we have is something called wobble pairing. So here, if you look at it, most of the third letter codes are not that relevant because they code for the same amino acid. So what happens in translation is that because of wobble position, the first two positions are more important to be matched. And the third one, even if it's not a perfect match, sometimes the tRNA can bind there and give rise to a modified protein or same protein, right? So this is called a wobble position. So that's slightly less important when the anticodon is binding. So what happens here is basically the tRNA. So this is the anticodon end. This is the other end, which is bound to basically a peptide chain. This is a tRNA that is bound to a single peptide. So what will happen is this, when these two come together, so there'll be a release of a OH. So a H group will come here, and basically a tRNA will be released, and the polypeptide chain will be extended. So that's how basically protein polypeptides are formed. Now here we see basically a ribosome, which basically is made up of rRNA sequence and proteins. So this is prokaryotic ribosome, this is eukaryotic ribosome. You can see that both of them have similar kind of structure, whereas they are made up of different, slightly different components, right? And this S here denotes Svedberg unit. So this is basically a substitute for the mass. So when you're centrifuging it, where does it separate out? That determines the S unit that we find here. So it basically is like some sort of molecular mass. So the higher means bigger, right? So there's basically a structure of a ribosome, which is used in protein synthesis, and is basically inside the cell showing where all ribosome is present. So the ones that are, so there are some that are free, there are some that are attached to the endo ER, right? So this is again the structure of RNA in the last subunit of bacterial ribosome, as shown by X-ray crystallography. So ribosome is basically what is an enzyme which uh, is composed of rRNA, which basically helps in protein synthesis. So here we see again the ribosome structure. These are the different subunits. These are the overlapping where it's overlapping. So here we have a large ribosomal subunit and a small ribosomal subunit, and they have three sites. So the A site, B site, and E site, also called the exit site. So A site is where the tRNA would enter, and E site is where it would exit. So this is how the process of translation occurs. So you have a polypeptide chain that is growing. You have an mRNA strand that is here, or messenger RNA. Then you have the binding of this ribosome. Then what happens? You have, based on this code that is present here. You have a complementary anticodon that is binding here, bringing in another amino acid. Then what will happen is the last ribosomal subunit will move, will basically sever this bond and basically transfer this to the second place. Then the small ribosomal subunit would move further, and then this previous tRNA molecule which was present will be thrown out. Then step one again gets repeated. We have another tRNA molecule binding to the next next codon, bringing in another nuclear amino acid, and basically that's how then the ribosome will move, and that's how basically the mRNA is translated into a protein. So what we have is, again, this is another advantage of having an RNA is basically you can have a polyribosome complex. So one RNA can lead to multiple copies of the same protein because multiple ribosomes can attach to it. So here is one ribosome which just started attaching and will start making a polypeptide chain. And here is another ribosome which is almost about to uh, separate from this mRNA strand because it is done translating the protein. And this is a photo of the same in the cell. So basically, we have a lot of inhibitors or a lot of molecules that are used as drug, which basically inhibit protein or RNA synthesis, and that's how they basically get rid of bacteria and other infections that we have. So, for example, tetracycline blocks the binding of amino acid tRNA to the A site of ribosome, and that's how it disrupts the translation. Again, another inhibitor which works only on bacteria is rifamycin because it blocks the initiation of RNA chains by binding to RNA polymerase, bacterial RNA polymerase, and prevents RNA synthesis. Then we have other molecules that act both on bacteria and eukaryotes, such as actinomycin D, which basically binds to the DNA and blocks the movement of RNA polymerase. So it prevents RNA synthesis. Next, we have things that work on eukaryotes, but not on bacteria, such as cycloheximide, which blocks the translocation reaction on ribosomes, and alpha amanitin, which basically blocks mRNA synthesis by binding to RNA polymerase 2, which is found in eukaryotic cells. So the next thing that we see is basically something called the co-translational folding of a protein. So I took all these images from such slides. So basically, some of these are not that great. So like some of these have annotations which have been translated, so it's not that great. So just excuse that. Sorry. So we have a mRNA and we have ribosome, which is basically producing a protein. And while the protein is being produced, as the domains are getting produced, we also see that the proteins start folding. So basically, the translation and folding of a protein go hand in hand many times in the to produce a protein or polypeptide chain. Right. So that's it for the last previous chapter. Does anyone have any questions? Okay, so we'll move on to lecture 15, which was on gene regulation. So first we'll talk about what is gene regulation and why do we need it. So some genes, as we know, are always on. So this is known as constitutive expression. Then if there are certain changes in the environment, there might be some of these genes which are always expressed to be stopped, and maybe some genes which are not expressed to be started, and so on. So these changes, these regulations, for example, like if you have a bacteria which is in a Petri plate where there's no glucose, uh, the addition of glucose will lead to basically expression of genes that can decompose the glucose in the bacteria or take up glucose by the bacteria, right? So those genes need to be upregulated. Now, if you change the source from glucose to, let's say, another sugars such as sucrose. So the bacteria would now need to upregulate genes that help it digest sucrose while downregulating genes that help it digest glucose, because glucose is no longer available that much. What we have here is more sucrose. Right. Then there are other stages, such as during the development of cell or the development of a organism, where basically there's cell differentiation that's occurring. So basically, the cell divides into two parts that are not similar in function. So in that case, one cell needs to perform certain functions, express certain proteins. The other cell needs to perform certain other functions and express certain other proteins. So that's also another case where we need gene regulation, right? So here basically we show two cells. On the left hand side, we have the bacterial cell. On the right hand side, we have the eukaryotic cell. So a bacterial cell has no concept of a nucleus. So the DNA is present here. It's present as a loose strand, right? So the DNA in bacteria is very loosely uh, bound. And that's why the DNA can be easily transcribed into mRNA, which can be used to produce protein, all happening in the same compartment, which is the plasma membrane. Whereas in a eukaryote, what happens is the DNA is very tightly bound due to presence of histone proteins and so on. So what happens is the gene is generally in a inactive state, right? So for bacterial genes, what is happening is genes are generally in the active state. So to regulate the function of a gene, it needs to be repressed. Whereas in eukaryotic cell, what happens is since the DNA is bound very strongly, very tightly, so the genes are generally present in repressed state, and they need to be activated. So what happens in eukaryotic cells is that so the gene opens up slowly and basically produces. So the DNA is transcribed to produce initially what is called the pre-mRNA, which then becomes the mature mRNA with the addition of a cap, in the addition of a polyadenine, poly-A tail. Then basically this structure, this mature RNA can go out of the nuclear membrane where it can attach to ribosomes and translate into proteins. Right. So here are some example rates of transcriptions that are found in different organisms and rates of translation. So as you see that the rate of transcription and translation is very high in E. coli, which is a bacteria compared to so the rates of transcription are comparable, where the rate of translation is very high in E. coli compared to eukaryotic organisms such as mouse. This is probably because of the nuclear barrier and the additional uh, capping and annihilation that is needed to from the mature mRNA. So here is an example of what we see. So we have a oocyte and a sperm, which basically uh, join together to form the zygote, which then divides to form what is called the morula, then blastocysts, and uh, other different human fetal forms. Then basically these this basically develops into an organism. So what we have is essentially one organism that is getting derived from this single cell, which is the zygote, which is formed by the combination of the oocyte and the sperm. So but what we see is in the human fetus, even in the human organism, we have different kinds of cells, many different kinds of cells. So we have neural cells that are present in the nervous system. We have circulatory cells, we have blood cells, we have immune cells, we have skin cells, and so on. So all of these come from the same cell. So they should have the same DNA. But again, they perform different functions. Therefore, there should be some amount of regulation that is happening. Right. So here again, we see that all of these different kinds of cells, the fibroblast cells, epithelial cells, neurons, stem cells, muscle cells, all of them arise from the same cell, which is initially the zygote. Right. So how does this differentiation occur? So people had two different ideas initially. So one was that there are different components of the DNA that are passed on to different cells. So for example, these cells have only these parts of the DNA, these cells have these parts of the DNA. And when they break into four cells, all of them have different parts of the DNA, and that's how they are expressing different proteins. And basically, that's how they are giving rise to different kinds of cells, different kinds of functions. The other theory is that uh, all of the DNA is copied into all of these, but we see that only one of them is expressed in each of these cells. So the second part is actually what is happening. It also shows us that the DNA that is found in all the cells is the same because the DNA is preserved, the DNA is not left out. What is happening is even in the presence of all of the DNA, only part of it is getting transcribed, and which part gets transcribed depends on the cell that you are in, right? Or the function of the cell that you are looking at. So this is basically an overview of gene regulation in eukaryotic cells. So what happens is we have introns and exons that are present in the DNA. They're transcribed into pre-mRNA. Then the pre-mRNA basically by splicing removes the introns. And there's an addition of a cap on this end and a poly A tail. So AAA. And then this basically is exported outside where basically ribosome binds to it and translation occurs to give rise to a polypeptide sequence of amino acids, which is called a protein. So there can be multiple steps, multiple things that are going on here, which can give rise to gene regulation. So the first thing is there can be differential gene transcription. So DNA methylation is one way where transcription of certain parts of the DNA stop. So you don't have any transcription, so no mRNA is produced. There can be selective mRNA processing. So only some parts are spliced, only some RNA are processed, others are not processed to go out. Right. There can be selective translation where after the RNA comes out, it sometimes disintegrates or there's another chemical, another molecule that basically binds to the RNA and prevents its translation. And then there can be something called protein modification, where basically after the protein is formed or when the protein is being formed, there's some modification that's happening to it. Right. So there can also be other proteins. So for example, these proteins called transcription factors basically come and bind to these uh, sequences in DNA present called enhancer sequences, which basically help upregulate or downregulate the transcription of certain proteins. So this is what basically will happen. So the transcription factor will bind to an enhancer, then it will basically form a protein complex, which basically binds to the RNA polymerase and starts the transcription of a certain gene. So proteins can also regulate the transcription in eukaryotic cells by the virtue of binding to the DNA as like transcription factors and so on. Right. So now we have a modified form of the central dogma where we have other arrows. So we have the DNA, which can be transcribed into the RNA which can be translated into different kinds of proteins based on how it is spliced. Then different kinds of proteins can basically come and bind to the RNA to produce certain functions. For example, uh, yeah, polymerase and all that bind to RNA to basically uh, yeah. So uh, ribosome is mostly made of RNA. Yeah, so we have some proteins that basically bind to RNA to form complexes, as we learn about in further lectures. So when we look at microRNAs, then we have proteins which are, let's say, transcription factors, which can basically bind to the DNA and stop or increase or decrease the transcription of RNA from DNA. Then we have RNA, which can basically give rise to non-coding RNAs or microRNAs. So in the next lecture, we look into non-coding RNAs. Does anyone have any questions till now? Okay. So as we know before, the human genome has basically a lot of DNA base pairs. Only 1.5 to 2% of that codes for protein, and only two-thirds of the gene, which is around 70%, is transcribed into mRNA, into RNA. So basically, this 1.5 to 2% of DNA is transcribed into mRNA, which eventually gives rise to a polypeptide. This two-third, which is whatever else is left, is not generally transcribed into mRNA, but it's transcribed into non-coding RNA. So what are these non-coding RNAs that are made of? So non-coding RNAs are basically made of different kinds. So for example, we have the tRNA and the rRNA. So the rRNA, tRNA basically help in translation protein, right? This is present in ribosomes, and this is present in uh, tRNA basically binds to mRNA to basically help in translation, right? It acts as an adapter. Then we have small nuclear RNAs, which are basically around, let's say, 150 nucleotides. And they are basically regulators of transcription factors. They regulate transcription factors. Then we have microRNAs or miRNAs, which are 20 to 24 nucleotides long. And they are basically single-stranded RNA molecules, which basically form a hairpin structure, which we'll look into further. And they basically help in post-transcriptional activities or post-transcriptional regulation. Then we have long non-coding RNAs, which can be around 200 nucleotides, which basically regulate microRNA and uh, small interfering RNA. Right. They also help in post-transcriptional regulation, and they are basically also used for chromatin modification. Then we have these small interfering RNAs, which are again around 20 to 24 nucleotides long. And these basically also help in post-transcriptional activities. The important thing to note here is that small interfering RNAs are generally exogenous, and they are also called as RNAi or RNA interference. So exogenous means they are coming from out of the cell. So this leads to a wide range of biotech applications, which we'll talk about in a bit. So next we have this organism called C. elegans, which is generally used as a model system in many of the developmental studies. So it has different stages of life, including an embryo from where it starts. Then it has a L1 phase, followed by a L2 phase, followed by a L3 phase, followed by a L4 phase, followed by a adult phase, in which again gives rise to the embryo. Right. Now, this is where microRNAs were basically first discovered. So what happens here is this transition from one stage to the next is what is encoded by microRNAs. So we have this microRNA here called LIN4, which basically is a microRNA, which helps downregulates this other protein called lin14, which basically leads to a change of phase from L1 to L2. Similarly, here from L4 to adult, we have another miRNA called LED7, which again inhibits the production of Lin41, which is another gene, which basically leads to the change from L4 to adult. So if we look here, so as the C. elegans progresses from L1 to L2, what we see is that the Lin4 RNA, Lin4 miRNA, 7 miRNA, basically increases in production. So what this leads to is a sharp decline in Lin14. The Lin14 translation is what it stops. So what happens is the Lin14 protein stops producing, and then the C. elegans basically progresses into the next stage of life, which is L2. Similarly, from L4 to adult, we have this LET7 miRNA microRNA, which is triggered, which basically reduces the production of Lin41, which leads to the adult phase. We see that the very strong switches that are present here. So this is a very fast change. So the cells are initially present in L1 phase, then suddenly, in a very short span of time, all of them go into the L2 phase, and so on. So this is a very small, fast kind of switch that is done by microRNA, and it serves as a regulatory RNA sequence, regulating the production of different proteins in the cell. Right. So here we see an example of how microRNA, small interference RNA, siRNA, small interfering RNA can lead to silencing or gene, basically modification of translation and transcription of other genes. So generally, the siRNA can have a hairpin structure or a double-stranded RNA. The microRNA will almost always have a hairpin structure. Then there's this molecule called dicer, which binds to it and then cuts it. Then this basically double-stranded thing breaks down into a single strand, and it binds to other proteins to form what is called the RISC complex. Then this RISC complex binds to the DNA or the mRNA, sorry, binds to the mRNA, and basically it can carry out gene silencing via a lot of different things. So what it can do is for microRNAs. So this thing is valid for siRNA. For microRNA, what happens is the RISC complex basically binds to mRNA. And basically, it can do a lot of different things. So mRNA, as you know, has basically a cap, a poly A tail. So what it can do is it can do stop ribosomal initiation. It can prevent a ribosome from binding to this. The next thing it can do is it can do cap binding, which is basically it will remove the cap or go bind near the cap, preventing translation. Then it can do prevent elongation. So if it goes and binds, the RISC complex goes and binds someplace here, the elongation of the protein molecule is prevented, and thus the full protein is not transcribed. And the other thing it can do is deadenylation. Which is it removes of the poly A tail, and thus the translation of the protein is not possible. Right. Now, small interfering RNAs, siRNAs, as I talked before, led to something called RNA interference. So what this is basically these two people, Craig Mello and Andrew Fryer, got the Nobel Prize in 2006 because of showing this. So what they did is basically if you have a double-stranded RNA that you're introducing into a C. elegans, it leads to the basically silencing of a gene or a muscle protein that is leading to a twitching in the worm. And they show that if you do this by just using a single-stranded RNA, it's not possible. It's only possible if you do it using a double-stranded RNA because the dicer molecule needs a double-stranded RNA or a hairpin loop to attach. Right. So basically this paved the way for different kinds of things and help us extend our knowledge. First of all, by we can basically regulate the expression of certain genes that we uh, basically might want to up and down regulate. So, for example, if you know that a disease is caused by a certain gene, what you can do is you can have RNA treatments or small RNA strands which can be introduced into the patient, which prevent the transcription or the translation of that protein that's leading to the problem. The next thing is that we can silence genes to understand what function they were carrying. So, for example, here by silencing this gene, what they figured out was this gene did something which led to the twitching. Right. Now, if by silencing this gene and another gene, they can stop the twitching, they know that these two genes are related to each other. These two genes perform antagonistic functions. So basically, they can study gene function using these RNA tools. And that's how this knowledge has helped us. So that's it for the lectures. Mm, Does anyone have any questions from the lectures? If not, we'll go forward and discuss practice assignment 3 for week number 3. Okay. So the first question is, what are the 4 building blocks of the cell? A, sugar, fatty acid, amino acid, nucleotides. B, glucose, cholesterol, phospholipid, tryptophan. C, proteins, amino acid, tRNAs. D, nucleic acid, nucleotides, RNA polymerase. So the 4 building blocks of the cell, as we talked about before, is option A, sugars, fatty acids, amino acids, and nucleotides. Sugars give rise to polysaccharides. Fatty acid gives rise to lipids. Amino acid gives rise to proteins, and nucleotides give rise to DNA and RNA. Polynucleotides, right? Nucleic acids. The second question is, which of the following is likely to be untrue? Amino acids come together via a dehydration reaction to make protein. Polysaccharides are broken down into smaller sugars by a hydrolysis reaction. Nucleic acids come together via a dehydration reaction to make a nucleic acid string. Polysaccharides are broken down into smaller sugars via a dehydration reaction. Which of these is likely to be untrue? So amino acids, as we know, come together to form a peptide bond by a dehydration reaction. So a water molecule is left. So this is correct. This is not the So this is a correct statement. This is not untrue. Next, we have polysaccharides that are broken down into smaller sugars by a hydrolysis reaction. That might be untrue. If we just go back and check. Okay. So this thing is not true because if this was the case, then basically starch would disintegrate if you put it in water. Right. The third option is nucleic acids come together by a dehydration reaction to make a nucleic acid string. So that is likely to be true. Polysaccharides are broken down into smaller sugars by a dehydration reaction. So that is also. Okay. Let's take a look. Actually, polysaccharides are broken down by a by a polysaccharides are broken down by a hydrolysis reaction and not a dehydration reaction. A dehydration is how a polysaccharide is formed. So this thing is untrue. Polysaccharides are broken down by a dehydration reaction. That is what polysaccharide is formed by a dehydration reaction. It's broken down by a hydrolysis reaction. Okay. The third question is, what is the relative contribution of each molecule in a cell? So again, if we look at, so this is in terms of what is present more, so the constitutional components, we see that nucleic acids, protein, lipid. So we know protein is more compared to nucleic acid. So this is false. RNA greater than DNA, we know this is true. Protein greater than nucleic acid greater than lipid, this is true. All of the above will be false, B and C. So the answer will be this, option E. The next question is, which of these statements accurately reflects the differences between RNA and DNA? DNA is double-stranded, forming a double helix, while RNA is usually single-stranded. It forms a hairpin. The sugar, so this is correct. The sugar in DNA is deoxyribose, whereas RNA contains ribose. This is also correct. DNA uses the bases adenine, thymine, cytosine, and guanine, while RNA uses adenine, uracil, cytosine, and guanidine. Okay, so this is incorrect because this should be guanine, not guanidine. So A and B are correct. All of the above. This is incorrect. So the answer here will be this, A and B only. If you have any question, if you have any doubts, please feel free to stop me and ask. Which of the following is true? Starch is a mixture of amylose and amylopectin. This is true. In cellulose, the OH group on the sugar is alternating. So cellulose can be closely packed by multiple fibers joining together. This is true. This is why we discuss cellulose is harder to digest. Glycogen has an open structure with multiple branches, allowing it to be broken down easily. That is also true because glycogen is the immediate store of glucose in the cell, and that needs to be broken down to get energy. The answer here will be D, all of the above. Does anyone have any questions? Lipid membrane contains A, lipidated proteins, yes. B, cholesterol, unsaturated and saturated lipids, yes. C, nucleic acids. Lipid membrane does not contain nucleic acids. D, phospholipids. That is true. So all of the above will be false, and the answer here will be E, A, B, D. Okay. What are some of the ideas of the central dogma as Crick presented it? Information is contained in DNA that is transferred to RNA and then to proteins, but never from protein to DNA. This is true. Information is contained in DNA that is transferred to RNA and then to proteins, and also in the reverse direction. This is false because protein does not give rise to RNA or DNA. Proteins are the template for DNA. Again, this is false. Transcription is the process of information transfer from RNA to DNA. This first is called reverse transcription. So the answer here will be 7A. Information is transferred. So question number 8. Information is transferred from one molecule to another by serving as a template or blueprint for the new molecule. Which of these transfers are not allowed? As mentioned before, DNA to DNA is allowed. This is called self-replication. RNA to DNA is called reverse transcription. RNA to protein is called translation. And protein to protein is not allowed. So this is the answer. Protein cannot translate, or protein cannot produce another protein. mRNA is what produces protein. Question number 9. Why is RNA thought to be necessary as an intermediate between DNA and protein? Option A, multiple RNA copies may serve as templates to make proteins, providing the cell with more control over how many proteins need to be made. That is true. All RNA is inherited by the next generation. This is false. DNA and RNA are nearly identical, so RNA is likely a waste product of evolution. This is also false. So the answer here is A. RNA, as we mentioned before, RNA has a lot of make a lot of copies, which can then be used to provide a lot more control over how many proteins are to be made by the DNA. Right. Okay. So that's it. Does anyone have any questions or doubts? If not, I'll just go over all of these things once more in brief, and then we can stop. So basically, lecture 12 and 13, we talked about biomolecules, the essential elements of life, biological reactions, then building blocks of the cell, what is the composition of the cell. Then we talked about basic structures of DNA, basic units and structure of DNA, RNA, protein, the primary, tertiary, secondary, quaternary structure of a protein. Then basically we talked about polysaccharides and how they are formed. We talked about starch, glycogen, and cellulose. We talked about glycosaminoglycans, lipids, and then phospholipids and cell membranes. Then we talked about the chemical synthesis of hormones. So basically lipids like cholesterol and all that at the leading point of our hormones, such as cortisol and all. And we talked about how chemically they are synthesized from other sources. Then we talked about the central dogma, which was proposed by Crick. And basically, this is the central dogma that we talked about. Then what kind of transfer of information is allowed, what is not. Then what is the benefit of having RNA as an intermediary? Then how is RNA transcribed? Then RNA polymerases and the different kinds in eukaryotic and prokaryotic cells. Then we talked about the presence of introns and exons in genes and how they lead to different kinds of RNA sequences. Then we talked about the different kinds of RNAs that are produced in the cells. Then we talked about the translation process, making protein from RNA. We talked about the genetic code, the tRNA molecule, and how the tRNA molecule basically leads to polypeptide chain extension, and the ribosome, which helps in translation. Basically, how the process of translation occurs using ribosome, tRNA, and mRNA. Sorry, tRNA and mRNA. We talked about structures such as polyribosome, then how we can inhibit or protein or RNA synthesis, and how these are used to basically make drugs for bacterial or eukaryotic infections. Then we talked about the co-translational folding of a protein. Then we talked about gene regulation, why it is important, how it happens in prokaryotic and eukaryotic cells. Then what are the rates of transcription and translation in different cells. Basically, how gene regulation helps in developmental growth and cell differentiation. Then we talked about what different kinds of gene regulation occur in eukaryotes. We talked about transcription factors and how they modulate gene expression. Then we talked about in the next lecture non-coding RNAs. So 70% of the gene that is transcribed produces non-coding RNA. So what are different kinds of non-coding RNA? Then we talked about how microRNA was discovered in C. elegans and how it leads to gene regulation. Then the mechanism of gene regulation used in microRNA and SI, small interfering RNA, siRNA. Then basically how this Mello and Fire developed the process of RNA interference, which basically leads us to a lot of interesting biotech applications. And then we discuss questions. So does anyone have any doubts regarding any of this? Okay. If there are no doubts, we'll stop here. We'll meet again next week, same time. thank you for attending the session, and I hope you will attend the session, other sessions. Please let me know if you have any questions or any doubts. Please also you can unmute yourself, ask questions in the chat box, or you can basically ask questions on the discussion forum also. So if I'll just wait for 5 minutes if anyone has any doubts. Otherwise, we'll stop here. Okay, if no one has any doubts, we'll stop here. Thank you for attending. We'll meet again next week. Bye.